Hello everyone. Today we're going to talk about variability in scientific data. And variability, how data is spread out, is just a way of describing how data points differ from one another within a data set. When I say within a data set, what we mean here is these are the different changes to your independent variables, but within a data set, we can see that plants might grow at different levels. So what we call this in our experiment inside the quad planters, it would be like trial 1, trial 2, trial 3, or trial 4, or plant A, B, C, and D within a data set. In this case, this is 1% fertilizer given to these plants. And as we can see that overall, just from this visual, that plants that are given 1% fertilizer grow better. But let's look more closely at why this matters. So why does variability matter? When we look only at averages, we get a general idea of results, but we miss out on the details. So variability shows what's happening behind the average, how consistent data is, and if there might be other factors affecting the results. In short, it gives us a richer picture of what's going on. So let's look at this first graph. Here we can see total averages of plant height at each water level, and then it has a trend line to show how as we water the plant more, the plant grows higher and in average. Now keep in mind that these dots represent an average of all the plants that were grown in a quad planter. This gives us a clear idea of how the plant growth changes, and we can see the upward trend. That's why we have a line here. However, using just the average with the trend line hides a lot of the details of the experiment. We don't see the range of growth for each of the water levels, and we can't tell if there's any outliers or unusual results because everything is just packaged within each dot. So while this is a good summary, it doesn't tell us the whole story. Now let's look at the second graph, which includes data from all 4 trials at each water level. Here we can get a much better picture of what happened in each group. We can see that for lower water levels, the plants generally had less growth, all right? But there's a lot less variability. And at higher water levels, the growth varies a lot more. Some plants grew a lot, while some didn't grow at all or grew very little. This tells us that at higher water levels, other factors like maybe light or soil nutrients might have influenced the growth more. By looking at all the data points, we can see that the trend line isn't always the whole story. Including all the trials helps us to understand the variability and give a richer picture of how the plants respond to different water amounts. So how can this help you to show a better understanding of your presentation? Well, first you want to make a scatter plot graph that has a trend line like the one that we did in class. And then after that, you should make sure that you put some type of comparison between the data points that have more spread and that are less spread. In this case, I was able to compare plants that got more water versus plants that got less water, but you might just compare 2 different data sets. For example, you might just compare like the 10 volume of water versus the 50, and maybe you know why something happened that made the data more spread. So keep in mind that this is not something you have to do for this lab foldable. It's something in the appendix that you'll discuss as a way to show an extension. And if you need any help, feel free to have a conversation with someone.